What is going on, guys? Welcome back to our GCSE psychology journey, and in this video we're going to be looking at an introduction to neuropsychology. Let's get on it. So starting off with, let's get a better understanding of what cognitive neuroscience is. So what is cognitive neuroscience? Cognitive neuroscience is the scientific study of how brain structures and brain function are linked to cognitive processes, things such as memory, attention, perception, language, thinking, and decision making. So it combines two key things: cognitive psychology, which is how we think, and neuroscience, which is how the brain works biologically. So structure and function of the brain relates to behavior. So different structures of our brain are responsible for different behaviors. And this supports the idea of localization of function, i.e. different areas of our brain deals with different functions on how our body reacts and how we think and how we process information. An example of this is how our frontal lobe is heavily responsible for our decision making. It's responsible for our personality. It's also known as the seat of the soul. And our motor cortex is responsible and controls our bodily movements. Structure and function of the brain relating to cognition. So cognition refers to mental processes, things such as memory, language, and thinking. Cognitive neuroscience shows us that things such as like our hippocampus is heavily involved when it comes to memory formation, and what we can see highlighted here on our image is the hippocampus. Broca's area is heavily involved in speech production. Wernicke's area is heavily involved in things such as language comprehension, and we do see that in some of the previous videos as well, so if you guys go back and make sure that you have a look at those. An example could be that somebody who has damage to their hippocampus may suffer from severe memory problems. Damage to Broca's area can cause difficulty in producing speech. Cognitive neuroscience helps us understand mental disorders by examining abnormal brain activity or structures. Neurological damage. Neurological damage really helps us to focus in on the importance of localization and understanding localization when it comes to the brain. So localization of function means that specific areas of the brain are responsible for particular behaviors or processes. So for example, our motor cortex controlling our voluntary movement, Broca's area again speech production, Wernicke's area again language comprehension. And damage to the frontal lobe, for example in what we saw with the Phineas Gage case, showed personality and behavior can be linked directly to specific areas. And people who suffer from things like strokes. So a stroke occurs when the blood supply to a part of the brain is cut off, usually by something like a clot. And the effect depends on the area of the brain that is damaged. So for example, damage to the motor cortex can lead to paralysis on the opposite side of the body. Damage to Broca's area, you may suffer from Broca's aphasia, which is slow, effortful speech. Damage to somewhere like the occipital lobe, which is at the rear of the brain, can lead to vision problems. Effects of neurological damage on motor ability. So damage to areas involved in movement can cause paralysis, so the inability to move muscles, loss of coordination. So usually when we get damage to our cerebellum, which is that little mini brain on the back of our brain, now the motor cortex controls the opposite side of the body, so it's contralateral control. So damage to the left affects the right side and vice versa. What about the effects of neurological damage on behavior? So damage can affect personality, emotions, and decision making. Frontal lobe damage, so damage towards the front of the brain, damage at the front of a brain, can lead to impulsivity, poor judgment, and significant personality changes, as we saw with the Phineas Gage case. Damage to our amygdala, which we can see here highlighted on our illustration, which is that small cluster of nuclei, can lead to a reduced fear response. Prefrontal cortex damage can lead to difficulty planning and controlling our behavior. Scanning techniques to identify brain function. So how can we identify these areas that are either damaged or abnormal? So there are different scanning techniques. Brain scanning techniques allow psychologists and neuroscientists to study the structure and function of the brain. They're often used to investigate localization of function, neurological damage, and also disorders. Here we have a functional magnetic resonance imaging scan image, or an fMRI. And this detects changes in blood flow to show active brain regions. So by seeing what is highlighted, we can see which areas of the brain are active. The strengths of this method is that it has high spatial resolution and is non-invasive. So spatial resolution means that it can accurately identify the areas of the brain that are active when the brain is reacting to a stimuli. Limitations: it can be quite expensive and we have poor temporal resolution. Now, of course, we have the word temporal when it comes to our temporal lobe, but in this regard, it means the time the scan can pick up a reaction to a specific thing. So poor temporal resolution, so it doesn't react automatically. Also, it can be quite uncomfortable for some participants to get an MRI scan because it can be quite claustrophobic, quite closed off. So with a CT or CAT scan, the brain is examined by taking a large number of X-rays of it. Each image is a slice through or cross-section of the brain. So the strengths of this is that it's useful for revealing abnormal structures, things like tumours or structural damage. It has very high quality images, so high quality images are produced, more so than a traditional X-ray. Weaknesses is that it requires more radiation than traditional X-rays, so therefore you can't use it very often. And it only provides structural information, not activity, as with other scans like the fMRI scan that we just saw. What about a PET scan or positron emission tomography? So this uses radioactive tracers to study metabolic processes and neurotransmitters. The strength of this is that it can track biochemical activity. Limitation is that it's very invasive, it's expensive, and it has a very limited use due to the use of radiation. Let's have a look at Tulving's gold memory study. So the aim of this study was to investigate how episodic memory, or memory for a personal experience, and semantic memory, memory of facts and figures, are stored in the brain and whether they involve different processes. So the method was Tulving carried out a case study of a patient known as KC who had severe memory problems after a motorcycle accident. The word gold was used to test different types of memory. KC was asked questions that tested his semantic memory, so what is gold, and his episodic memory, have you ever seen gold, or when did you last see gold? Tulving compared KC's responses to those of people without brain damage. What were his findings? So he found that KC could explain what gold is, so it's a precious metal, so that means that his semantic memory was intact. However, he could not recall any personal experiences involving gold, so his episodic memory had been impaired. And this showed that episodic and semantic memory are separate systems, that brain damage can affect one type of long-term memory, but not the other. So let's evaluate that. So what are the strengths of this study? Well, it provided strong support for the distinction between episodic and semantic memory. It was a highly detailed level of data from a real patient. It helped improve understanding of memory and brain localization. What about the weaknesses? Well, as with any case study, findings cannot be generalized to everyone because it is a very limited study focused on one individual. Brain damage was extensive, so it's difficult to know exactly which brain area caused the memory loss. We can't establish cause and effect because there are no experimental controls as we would have in something like a lab study. Alright guys, hopefully that gives you a better understanding of some of those scanning techniques and also of Tulving's gold memory study. Make sure that you guys watch over the video, make your notes, pause the video where you need to. If you haven't already, make sure you like and subscribe. I don't know why you haven't if you haven't already, but I will catch you guys in the next one. We'll continue our journey. Catch you guys then. Peace.